Maybe Rakesh Kumar. I can see S. Rakesh Kumar. If you can unmute yourself and tell me, how did you choose your MD thesis topic?
I chose my MD thesis topic because in my medical college there was particular mortality related to suicidal death of farmers
Okay.
consuming OP poisons and herbicide poisons. And I thought I could make a difference by studying this topic.
Okay. So did you choose it or did your guide choose the topic?
I proposed the idea and he proposed how to go forward.
Yes. So that's a very good way of choosing a research project, as we will see later. So I don't know, because we can't take head count here, but when I do these classes before last few years, it's always been that 95% of the students say that they have chosen their MD thesis topic based on what their guide said. And they hardly had any liberty to choose any topic. So you are fortunate, Rakesh, I must say, and credit goes to you for your keen observation. Thank you. If you look at the difference between research and discovery, discovery is like Charles Darwin saw something and we thought of it, or we can just discover something out of blue. Whatever we find for the first time, we say we have discovered it. But research is not that easy. Research is it's a very scholarly or a scientific activity. It needs very close, careful study in an orderly fashion to discover new facts or new information. So it cannot be done in a hurry. It needs time, energy, money, everything. So the question arises, some of you always think, why is this institution asking us to do this research project? Why should I do it? I am only going to see patients in future. So one of the criteria for students is because it's a requirement for your training. So you do it as a burden, as a compulsion to fulfill the requirement of your training program. As you did in MD, some of you might be doing that also in the DM. Then for faculty, requirement for promotions. And either you, according to MCI, you need to have X number of publications, and if you don't have it, you are not qualified as a teacher. But there are others who enjoy doing it, who have that questioning mind, who have quest for knowledge, who want to know why is this happening. And there are some others who do it to gain better status in the society, because people who publish are better known in the scientific circles. Even your institution gives you much more importance if you really publish and you are a researcher who publishes a lot. So it gives you a better status in the institution itself. And for the medical school, it helps in recognition of your college or your institution across the world, because nowadays everything is what is your institution contributing. And one of the areas in which contribution is counted is research publication. Other is obviously patent, how well you do patient care. And it's been very well said by a previous director of NIH that institutions which are well known across the world are also well known for their research contribution. Because when you do research, you have a questioning mind, and that makes you even question things in patient care, in patient diagnosis. You make better patient diagnosis, better patient management, and these two in turn lead to better teaching. So if you see good institution across the world, they would probably be good in all the three areas. And for the society, why does the society want us to do research? Because they want us to develop better drugs for them. They want us to develop better diagnostic tests so that the diseases can be diagnosed much earlier. And for a country like ours, we also need to have research on how to reduce costs of medical care. And last but not the least, actually, we don't want diseases. We want good health. So how can we have interventions which can prevent development of disease and improve our health indices? So that is what the society is looking at. So let's see what are the various steps when you want to do research. The most important and the foremost is choosing the right research question. Once you have the right question, you also need to develop a protocol on how to answer that research question. And then once you've developed a protocol, you need to execute. The execution means you collect the data during the time you're doing the study. And once you've collected the data, you need to make sense of it. To make the sense of it, you need to analyze your data and find out what is this data telling you. And last but not the least is to draw and disseminate your conclusions. If you have done it and you haven't told anybody, it is as good as you haven't done it. So what is a research question? A research question is nothing, but it's an uncertainty about something, that we're not sure about something. Actually, in the population, because whenever we talk of the whole population, that the investigator or you want to resolve by making measurements or some study on your study subjects, either by just seeing them, like we do it in an observational study, or we interfere with the natural course, like you do an interventional study. But all interventional studies are done in a planned manner. It's not a haphazard intervention. It is a planned intervention. So this is what actually a research question is. So a research question can be as simple as that. What causes dental caries? Can we do something about dental caries? Or it can be even when you see our patients in the OPD are not satisfied. One reason could be that they have long waiting times. So how can we improve the patient satisfaction in our outpatient clinics? Or it could be that how can we help patients with high blood pressure who cannot take drugs which are currently available? Can we provide them some new drug? So as Rakesh had told us just in the beginning, that he thought of a research question by observing a phenomenon. He found that there were a lot of farmer suicides by using organophosphorus. So he did study on that. So you can have, if you're a keen observer, and all of you have done MBBS and MD, so you have already had 8 to 9 years of training, you may have made some observation during this period, and you may pursue that. Why is that observation happening? It could also be a practical problem in the field. Like we have patients who cannot afford standard of care. Can we provide them some cheaper therapies? Or can we repurpose drugs? The drugs which are used for other disease indications, but they have many other pathways which they affect. Can we use them? Because they have been used for long. They are much cheaper than the drugs which we are using. Or it could be that you're reading every day and you find something which is so fascinating and you would like to pursue it. Then sometimes what happens is, like COVID-19 came. So every funding agency is sending out requests in the newspaper, in the other areas, that please send requests, proposals to us, or research proposals on COVID-19. So you might think of a proposal on COVID-19 and send it to a funding agency, or you might do it just yourself. So you always have to think of an idea when you want to do research. Then for people like us who have been in the field for 30 years, we work on only very few focused areas. So whenever we do research, we always, with one project, you answer that question, but there are 10 more questions which come up during the period. So you build up in your previous experience and you gradually keep on answering one after another question. You also should be alert to new ideas, either when you're reading or when your friends are presenting journal clubs, because journal clubs are one place where you can really think of new ideas. Because most of the journal clubs which are presented in our most departments are very recent topics. They are state-of-the-art articles, and they would also throw up some questions. Alternatively, when you go and attend national meetings, their data is presented. Some of the data may look very different, and you might think, why are they finding this kind of a results? And you may like to answer that question. For radiologists or for interventional people who do intervention, a new technology comes, and then they want to compare. Like when the PET CT came, everybody is comparing how good is PET CT to detect residual malignancy vis-a-vis conventional CT. So whenever or people who are cardiologists, if you get a new stent, They will compare, is drug-eluting stent better than bare stents? Or if something else has come in, you can do a comparison study. So ideas can come from multiple sources. But you must have your imagination roaming. Then only you can imbibe those ideas and think of a research project. The most important thing is that the idea which you are thinking, is it really new? Because we really don't want to do research. That is, that everything is known, we are just doing the same thing over again. So how do you find out that it is really new? Like Dr. Ghoshal has just told you how to do literature search. So one thing is that do a good literature search using PubMed. You can also use other search engines. It's not that everything is indexed in PubMed. There are articles which are published outside PubMed. So you can use Google Scholar. You can do it on other, just put it in Google itself and search. You can look, generally it's a good idea to look up recent journals. And when you do search, don't do a very narrow search, because sometimes the articles are published in related areas. So do a little broad search and scan through those articles so that you know that nobody has published in that area. Generally, textbooks are not good for research ideas, but textbooks give you an overall idea about that subject. The other thing is to walk down to your mentor and ask that person, because that person has been in the field for 20 to 40 years. They know overall what is happening in that area. So they may be able to help you and tell you that, is it really new or not? Because we don't want to reinvent the wheel. And because what happens is that if we don't do a good search at this time, you plan your study, you finish your study, and then when you want to send for publication, they say, oh, it's been done 20 years ago. There's nothing new. And it never gets published. So it's better to work in the beginning than to feel sorry at the end. So when you plan any research question, in addition to it being a novel question, you also should be clear, what is the purpose of this research? Its fundamental concept, its main aim. Is it to help patient care? Is it to improve patient satisfaction? Is it that I'm just doing it for my own curiosity? Everything is good, but you need to be clear. Don't be ambiguous in that, because every research means a lot of time, energy is spent on it. So it must have some significance. After you found out that the research idea is novel, then we need to find out, is it feasible? Because I can't do, if suppose I want to see, is there a life on Mars? I know it's not feasible. I won't be able to do it. So I need to find out, is my idea feasible? And feasibility has multiple components, especially for a project which you do in a short period of time, that you have only 2 years to do a project. So you need to be sure that this project can be finished in time. You have equipment and chemicals available, because if you think that your department will order an equipment and then you will do the project, it may not finish in time. Then do you have the training and skill to do that thing, or your mentor or in your department that is available? If not, can your department collaborate with somebody to get that skill for you? Then do you really have the substrate? If you're working on patients, would you be getting adequate number of patients in that time frame or not? If you're working on animal model, would you be able to develop that animal model or not? Then do you have the money? Because no research can be done unless it's only questionnaire based without money. So your supervisor needs to have that kind of money which is needed for the project. And lastly, always think that if you were asked to participate in that study, would you like to participate or not? Then only do that study, because the ethics of research are the most important. We have some sources from where the students can get funding. ICMR has a scheme where they give you thesis grant for MD and DM. There is a separate scheme for Northeast region students. Then some institutions give grant for MD thesis and DM thesis. Our institution provides it through the intramural scheme. That is, your mentor can write an intramural scheme, and through the intramural grant, if they can get it, then a student's project can be funded through that. Some societies provide grants for student project, like our society gives grant of about 1 lakh, but these are all competitive grants. It's not that you sent it and you will always get it, because there are limited numbers. So Indian Journal of Rheumatology also used to give grants for undergraduates. Asia Pacific region gives grant. And for PhD students, some grants are available from DST Inspire. Now, once you've thought of a research idea, now you have to make it into a research plan. So your research question is for the whole population, but the study obviously cannot be done on a whole population. You will only be using a small sample. So here, your research question is a phenomenon of interest, but here you will be using certain variables to measure that phenomenon of interest. It's always good that your research plan has one primary question, because the primary question is the one on which you calculate your sample size. Other questions are always secondary. If you calculate your sample size based on 7 questions, then you have to take the largest sample size. So always have one primary question in your study. So now you've had your research question made, you now need to develop a protocol. So for protocol development, always you need to have a research hypothesis. A hypothesis is nothing but it's a logical supposition. Because whenever we read, after you've done a PubMed search, you have a reasonable guess or an educated conjecture that which provides a tentative explanation for the phenomenon which you're investigating. Like I might say that sugar causes caries. If I had a question, what causes caries tooth? I think children who eat a lot of sugar, candies and chocolates, and they are the ones who get. So I may say that chocolates may cause. So it always has a may, because I'm not sure. I'm just only making a guess. So a research hypothesis is like this. The excess salt in the soil may affect plant growth. A drug A may reduce diastolic blood pressure. But till you prove it, you're not sure. So then we convert it into a formalized hypothesis. Like if we are saying that a drug A may reduce diastolic blood pressure, we say if drug A is related to diastolic blood pressure, then the blood pressure will be expected to be lower after a person has received this drug than what blood pressure he had before it. So the independent variable here is the drug. The dependent variable is the blood pressure, because blood pressure is dependent on the drug which we will prescribe. So you need to have, this is called the PICO protocol. So PICO means P is for participant. Who is the patient or what problem is being addressed? Intervention. What is the intervention you are giving to the patient? Comparison. What are you comparing with? So what is the comparison group? Outcome is what outcome are you measuring? And you also need to decide what study design is the best for answering that question. So you decide on your tools and methods, you define your patient population very well, define very clear inclusion and exclusion criteria. To avoid any ambiguity, write it down. Define your primary endpoint, that is the primary question, and the secondary questions. Define your measurement tools. How would you measure it? If you're measuring blood pressure, would you measure by sphygmomanometer? Would you measure by electronic instrument? Would you do an intra-arterial blood pressure monitoring? So you need to be absolutely clearly defined. Would it be after 20 minutes of rest? Would it be in a standing position? Whatever. Define your sample size based on your primary question and define your statistical method a priori. They don't need to be done after you've done the study, because you know your question, you know your population, so you really know what statistics you need to use. Then you write up your synopsis and present in the department. Always present in the department to get feedback from people. And don't feel bad if you get a critical feedback, because the person who's giving you the criticism is actually helping you to improve. Rather, he's doing you a service by giving his critical input. So the usual problems which we face are that the research, it's too vague. It's absolutely inappropriate. So you need to write down the research question, specify it, use all these points, and then write your synopsis. It could be that it's not feasible. It's too broad. You want to do 100 things under the earth, which is not feasible to do in such a short time. So you specify your fewer variables. Sometimes we find that we want to, suppose I want to work on a genetic disease. I have said I will see all patients who have microcephaly with this. I have made it too stringent. And I know that in 2 years, I'm not going to get that many patients because my guide says, no, you won't get. So I can do is I can expand my inclusion criteria. I may just take all patients with microcephaly, or I can increase my recruitment time if I'm not in a course. Sometimes the methods are inadequate. Then you go and consult the experts and learn new skills because those methods are either obsolete or they cannot be done here. If you find that it is costing too much and your mentor says, sorry, I don't have money, then you need to either get cheaper alternative, use different outcome tools which are cheaper, or you try to generate a grant. But the most important thing is that unfortunately we find that the question which we have raised is actually not wrong. Then you need to restart from beginning. You have to modify your research question and start from scratch. And ethical uncertainty, obviously. You consult your ethics committee people if you are not certain that is it going to be ethical or not. But generally, ethics is nothing but morality in you. And you can always know, is it ethical to do this thing or not? So once you have developed your protocol, written it, because writing is most important, because there you spell out in black and white. Then you carry out your study and collect data. When you are carrying out study, always record the whole procedure in full detail. Maintain a logbook. Record your results absolutely truthfully, because it doesn't matter if the result is coming totally opposite to what you thought, because that is probably going to be much more revealing than what you thought if the same came. And record your results immediately. What people do is put it on a scrap of paper. Okay, I'll go to the hostel and then I will put it in my logbook. It gets lost and then you can't retrieve it. So it should be recorded as soon as you have made the observation. Record even those results which do not fit with your hypothesis or are not reproduced later. If you want to cut out, we all can do mistakes. If you want to cut out anything, just put a line over it so that it can be read, that what you had written before that and what you have corrected. And keep all your original records. If you are doing a study on ECG, all ECGs need to be stored. If you are doing radiology, keep all your X-rays, CT, MRs recorded. It is said that for any research project, 10 years we should keep the data. If you are in the lab, store all your gel pictures. You can store them in electronic versions because ECG also fades out. So take a picture of ECG, scan it, and store it. Similarly, for lab experiment, ELISA readings, anything. And transfer your data to a computer. Because so many people just keep it in their pen drives. And pen drive can get lost anytime. And then the whole data is lost. So keep it at 2 or 3 places. The other important points are that whenever you do anything, do a trial run. See, are you getting reproducible result or not? If it doesn't come, try to sort out the problem. Same in the lab. If you're running an assay, look at how much is the variability between assay run 2 times or assay run the same day 3 times. So inter and intra-assay variability is very, very important to see. Because if you find a minor difference between your control and the other group, it could be just because of this variability. And actually there is no difference in the 2 groups. And we must run all appropriate controls, be it for clinical study, be it for lab experiments. So I see most of you people write, we have taken age and gender matched controls. Actually, we never do matching. Matching means that you match one patient to one control. What we do is we are doing age and gender similar controls. So it's good to be truthful to say that we have just done age and gender similar. Like I work with children for my research. I am interested in juvenile arthritis. I am very honest. I tell them, sorry, I am not able to take children controls. I am taking young adult controls. So I take controls from 18 to 20 years. Sometimes the reviewer writes that maybe they cannot be compared. I say, sorry, it's not possible for me to have children controls. I'm not ethically able to get controls from children. Then once you have done your studies, you need to analyze the thing. Analysis should not be left totally to the end. Always do a regular review. Assess your progress. Discuss with your mentor your problems. Don't put it under the carpet. And then they will become a big issue at the end of 2 years. So every 2 months or 3 months, have a meeting with your mentor. And if there is a problem, you can always modify things. And modification is allowed everywhere. If you and your mentor are not able to sort out your problem, solicit help from colleagues and do a midterm presentation in your department. Like we have a 6 monthly to 8 monthly presentation in our department for all projects, so that we can give feedback if project is not working out. Maybe sometimes even the project totally needs to be changed. But it can be done rather than doing it 2 years, it's better to do it 8 months. So once you have done it, you need to draw your conclusions, and you will be having a full classes on biostatistics, how to analyze the result data and to draw conclusions out of it and disseminated the conclusions. So that's to summarize, plan and discuss with your guide when you're planning a research project. Have a very focused question. Don't think you can do everything under the earth in 2 years. You must have a well-defined protocol, which should be written, because if you don't write, then you are not clear. You must do honest recording of data using correct tools and have midterm reviews or even a more frequent reviews with your mentor. Do revision if needed, analyze, and then make a conclusion. But the most important thing after doing all that hard work is that it should come into public domain. We have spent public money, public time, because the government is paying us for the time we are spending in the institution. So we have a responsibility that the time we are spending into the research project, that should be put into public domain. So we need to write and send for publication. As is said, that a work not published is actually work not done. So if you have any questions, I can take up.